Okay, so today I will talk about introduce your second homework, which is the flow stretching and the DNA force extension relation. And I will, introduce what to do for this homework, the second homework. But before that, let me discuss this phenomenon of elastic objects. When you apply force on elastic objects like rubber, let me show it to you over here. Suppose you have this kind of material, which is a rubber, okay? And what the property of this rubber is that if you apply force to the ends of the rubber, depending on the amount of force you apply, the rubber extends, right? This is its equilibrium unextended original length, and when you apply the force, you see its length is larger now because I apply force. So if you continue this, that you will see that there is a relation between the force you apply to the end of the elastic objects and the amount of extension. There is this relation, and in fact you have seen this in Physics 101 class on discussing the springs, right? If you have a spring, for example, which is fixed at one end and it is free at the other end, then if this is, for example, the equilibrium length of the spring, equilibrium means there is no compression, no extension of on the spring. But the thing is, what you do is, if you apply a force on this end, what happens that the spring gets a little bit extended. If this is, let's say, x is equal to 0, which is the equilibrium position, right? Or let's say x is equal to L, which is L0, right? X is equal to L0, which is nothing but measured from here, which is nothing but the equilibrium length of the spring. But if you apply a force, what happens is that the spring will extend. Depending on the magnitude of the force, okay, this length, let's say delta L, will be larger or smaller depending on the amount of force. And we just studied this kind of relation between the force, the spring force, and the extension, and we call this Hooke's law, right? So we have seen that this force is just proportional to delta L. Larger the force, larger the extension, okay? Well, this means that F is just equal to some constant multiplied by this extension, right? If this L delta L represents the amount of extension from the equilibrium position, measured from the equilibrium position. And if you want to draw the force and extension graph, what you will get is that this kind of graph, a straight line, because of this relation, right? Because F is linearly proportional to length, and the proportionality constant being just, which is the spring constant K. Remember these from 101. And if you produce this kind of graph, what the slope of the straight line will give you, okay, this is the slope. Let's say y and x, y divided by x is the slope. Well, slope is nothing but K, right? You can just find this spring constant, that so-called spring constant, by just analyzing the data, drawing the straight line, baseline, and the just curve fitting, and calculating the slope will give you K. So, and this is the way that how you originally, so experimentally, find whether the spring you are using a stiff spring or a soft spring. If it is stiff, if it is a hard spring, K will be large, but if it is a soft spring, K will be small. And the unit for K is It is Newton per meter in SI unit system. So why I'm discussing this? Because I have just introduced you this biomolecule, which is called very important biomolecule, which is called DNA. And DNA, I introduced you last week. I said that it's kind of double helix. It's a molecule, right? It has some bonds between these double strands. This is one strand, this is the other strand, but these strands are just bonded together through some chemical bonds between these adenine, guanine, cytosine, thymine. And so what you usually find the DNA inside the cell, okay, and in the nucleus, is not like this elongated string. Usually DNA is just curled, okay? It is curled. In fact, it is winded. It is wound on a special type of protein which is called a disc protein, which is called histone. And DNA is just wounded. There are many histones. DNA is just wounded. I just told about very shortly in last week's introduction of nucleic acids. So there is always this kind of curious about this mechanical property of this DNA, and asking yourself that what happens if we just apply force to the end of DNA? What happens to it? So is it kind of like behaving like a spring that it will extend a little bit depending on the force you apply it, right? And if it's so, can we calculate just a spring constant, right? And these kind of physical properties, mechanical properties of DNA are very important in researches of these genetics researches, etc. Because the scientists are studying some forces during the copying of DNA, during the transcription, during etc. These there are many phenomenon events are going on with DNA. There are enzymes or some proteins are coming and they do some processes over DNA, etc. So these all involve some mechanical deformations, some mechanical actions, etc. Through these mechanical actions, if somehow we can measure the forces and also we can measure the extension, etc., we can just get some very valuable information, okay, in these genetics researches. So we will kind of try to mimic these operations on DNA by using simulation, okay, by using this optical T-Wizard simulation. So the other important thing, of course, is how are you going to do this? How are you going to do these manipulations on DNA? How are you going to just grab the ends of DNA and that stretch it and measure the extension, etc.? So of course, as you can guess, the optical T-Wizard is the device for this, and the optical T-Wizard can do such kind of operations on DNA, and it's very, very important operations. And this is why optical T-Wizard got Nobel Prize 3 years ago, because these operations will let the scientists get very, very much valuable information about the properties of DNA, etc., and development of genetics, etc. Okay, so that's being said, that's why we are discussing now today this homework, is that we will learn something about about the DNA. So this is the cover page again. As always, I will discuss your homeworks over the report template. And the first thing that after the cover page, you have to fill out this abstract part. You just write down what did you do and what was supposed to do in this experiment, and what kind of results that you get obtained, like just 3 sentences. And what is about the introduction? What about the introduction? That it says that this DNA force extension is studied through a process called flow stretching, and we will do this in the optical tweezer simulation. Well, we are not going to use optical tweezer simulation. We are going to use another simulation that I just put on the report template assignment page on Canvas. You will see there, and you can easily download it. It's again a Java program. It's pretty much like optical tweezer simulation. Even there is some optical tweezer device in that simulation also. It's very, very similar to the previous one. And on that simulation, we will use this property of the simulation that you can provide a flow between the glasses. We put everything in the glasses, between two glasses, the solution, and the solution assumingly contains biological biomolecules or some micro objects that you can trap with optical tweezer, and you can manipulate them. So we can always give some flow to this solution. So what this flow does on the objects that are inside the flow, they will be dragged, right? They will be dragged, but if you somehow catch them and still the flow is going on, you will feel some kind of drag force on the objects. Okay, so we can control in the simulation the flow, and we can measure the force, drag force, etc. Because there's always this drag force if you're inside the fluid, and if you somehow try to move some objects inside the fluid, let's say you do it on the summertime inside the pools or on the seaside, etc. You just dive on it, and you have something in your hand. Even your body will resist in motion inside the water because of the drag. Okay, so what the drag is, in fact, this drag is the resistance of the object's movement inside the fluids. Okay, so we will study these kind of things through these simulation, and this is the materials that we are going to use. So there are some preliminary questions before you begin working on the report. So you're supposed to answer these questions. I'm not going to discuss these. Just for example, the first one: what is the force versus extension curve look like for a rubber band? I just discussed with you that in the beginning of this live class. So right, okay. So what the second question is, what do you think that the force versus the extension curve for DNA might look like? Well, this is up to you. I mean, would you think that DNA can behave like a rubber? So if you don't think so, just try to give some reasoning in your mind, because you know what the DNA structure is. Okay, they're just molecules, some different types of molecule groups. They are bonded together, right? And if you think that if you apply a force, these bonds will just extend, why not the DNA acts like a rubber band? Okay, so think about this, and just the answer to this question is just there is no unique answer to that. It's about your imagination. Okay, if you think that their DNA will look like a rubber band or act like a rubber, then just sketch this DNA extension versus force, etc. What this question is about. Just trying to get your imagination or your expectation or your supposition, your assumption on the physical property of the DNA. So next, the procedure. Of course, you're going to download the this stretching DNA simulation, and then you will run it Let me run it. Okay, let me run it over here on my. So it's called the stretching DNA. Okay, stretching DNA simulation. I just run it. So already once, as soon as you run it, you will see this again, and there is optical tweezer over here. You see this is the microscope end, the objective end of the optical tweezer, and this is the spot point of the optical tweezer that you catch the objects. But also there is this thing, that there is this stringy thing. Well, you can just say that this is nothing but the DNA that we will work on. And supposing that at one end this DNA is fixed to the glass somewhere, a fixed point, a fixed object, there might be another optical tweezer over here. And well, in fact, this is what in reality happens. What the scientists do is that they just grab these two of these beds, the spheres. One end is being fixed, kept by an optical tweezer, and the other end is again attached to other sphere, and that sphere is just moved by moving the optical tweezer, okay, and so manipulating it. So as you just can guess that if somehow you attach the ends of this DNA molecule, one to the other is sphere, the other is fixed at anyway, you can always do this operation. That you see, you can extend it, right? You can extend by just moving one end by the help of an optical tweezer back and forth. So that's what they do. Okay, let me show a real video of what they have done in a similar setup. Let me show it. Okay, right over here. So I hope you see it now. Okay, let me just freeze the video. So this is a real experiment, that the video shot of real experiment. So what you see over here, let me run this. Okay, yeah, let me run this by using another player. This is not good. I will open it with QuickTime Player. Yeah. All right. Now, let me explain this. So what you see over here, these two spheres, right? And in fact, these are the pictures of real. This is in fact a video of a real lab setup that a scientist just had grabbed these two spheres. One is kept being fixed at this position by optical tweezer. The other one is also under the trap of another optical tweezer, but this one is movable, so he's moving it up and down. And what you don't see on this picture is that between these two spheres, in fact, there is DNA. There's a DNA molecule. One end is attached to this one, the other end is attached to this fixed one. So we are not able to see it because the DNA molecule is very, very thin. It's just, I think, 3 or 4 nanometers wide. Well, it can be long. I think over here it's is as large as like 16 micrometer length or so. The length is very long, but well, being very means that in terms of nanometers it's very long. But the DNA is very, very thin object, so you're not able to see under a microscope, a usual microscope. There is a DNA molecule between these two, but you are not able to see it. So let me run the video first. What's going on over here? Okay. You see, this one is just dragged by the help of optical tweezer and back and forth, up and down. So what's going on? what's going on, what you see on these graphs. Okay. All right. Let me stop the video over here. So at some distance, this setup has a device, measuring device, so that it just measures the force on one end on, for example, this microsphere. It is able to just measure that force, and what you see, this force at right, it is this position, the force to be 5.5 piconewton. You see on this, let me enlarge it a little bit, right, so you can see better. So I mean, this scientist has some devices so that he can measure it. So as you move further, as he moves further to another location, the force goes to 11.14 piconewtons, and he does it further and further. Eventually, the force he measures to be 17.4 piconewtons. So with this data, he gets some data, and also he get this distance, right? Because as he moves this open end up, the length of DNA is just extending, or he is also able to measure this extension length. So maybe you can do it yourself. You just know what these numbers are, and with this scale of this much of length bar being 5 micrometers, you can measure the distance between these two and this two end. So you can in fact do it yourself even. You can open just this video. I will just also put this video in the report page, in your Canvas page, so that you can download it and you can just run it and work on it or see it anyway. So you can produce force versus extension graph, and by this graph you will be able to, just by using this data over here, measure the spring constant of this particular DNA. Okay. Well, knowing the spring constant means that how much force, if I apply how much force, let's say if I apply this much force, and how much the DNA will extend, how long will it extend. So it gives you this information. Knowing the spring constant is very, very valuable. Okay, that being said, now I will just discuss what to do with this simulation. For this simulation, we won't use the optical diffuser, so we just turn it off. Okay. Once you turn it off, you see the motion of the sphere that DNA is attached to is going to this point, right? Why? Well, you might think that the DNA is just, it was stretched while the sphere was over here somewhere, but when you just release it, it will be pulled by the DNA backwards. Well, the sphere is doing this zigzag motion, because of the Brownian motion. And by the way, I want you to just, this is just a homework for you, another small homework. Just search what the Brownian motion is. Let me type it over here. Brownian motion. And this is a phenomenon that happens in fluids, to microparticles in fluids. Whenever you observe under microscope some microparticles of fluids, you will see that these objects will doing this kind of zigzag motion. For the time being, well, just search it and learn it. What is Brownian motion? So, but for the time being, just to make our measurements easier, we will just disable the Brownian motion for the time being. That by unchecking this crossbuck, enable Brownian motion. Okay. Now, the sphere is not doing this Brownian motion, zigzag motion, but you can just move it by grabbing it by using your mouse. Whenever you grab it at some position over here, you will be seeing that the DNA is just pulling the sphere towards this fixed end. Okay, and this is called the tack. Tack is over here. When you just move the sphere away from the tack, somewhere over here, and you release it, you will see this motion. Okay, because of the DNA is extended and it's trying to pull it back to the point. All right. Well, we will try to measure the position again of this sphere by using some tools over here on the side window. But how to do it? First of all, you have a ruler, a big ruler. When you just check it, you will be seeing this ruler. But this ruler can be the zero of the origin of the ruler can be relocated by using this setup of the optical tweezer in sideways, so you can just relocate it anywhere you want. Okay. And the other thing is that there is this position histogram that is also being used when you check it, as you have used it in your previous simulation. Whenever you check it, there will be this dashed line that crossing through this zero of the ruler, and this shows that the position measurements of the sphere will be done by using this origin. Okay, and this one, this window, through this window, you'll be able to do these measurements more accurately. Okay. So anyway, if you just grab this and move this dashed origin dashed line towards the position of the sphere, you will be able to see the position measurements right on this window. Okay, you have done it in your previous homework, so I'm not going to the details about this. And there are also these plus and minus to change the scale of your measurements, and there is also ruler over here. You can measure this bar, the location of the bar, that just measuring the location of the sphere with respect to this origin. That means this is the dashed line over here, this is the dashed line, this is the same line over here, and this is the location of the sphere with respect to this dashed line. Okay, so this is how you measure the location. Now you know how to measure it. Just initially what you do is you just bring the initial position or the zero of your tweezer to the point of this tack, this green tack. Okay, so you just you see that it's at the zero. That means your sphere is at the tack, but the tack is at also at zero. Okay, so at this point, what are you going to do? Well, you are going to run this fluid controls. As I said, we will give some fluid flow to this solution from left to right that you have done already in your previous simulation. So there is these controls that flow speed. If you give some flow speed, let's say, let me slide it toward right, so you see, observe what's going on. I just gave some flow speed, a uniform flow speed from left to right. Now it's 76 micron per second. So you see what happens. Because of the flow, the sphere will be dragged toward right. To some extent, of course. It just moved to some extent, but it stopped there at this extent. Why? Because the DNA is pulling the sphere in the backward direction, while at the same time the flow is dragging with some other force, with the opposite force to the right. When these two forces are equal to each other, the sphere just stops its motion, right? And you might be curious that whether you can measure these forces. Yes, you just check the boxes, the DNA force, and also the drag force. Okay, and show the force values. So if you also check the force values, the force that shows to the right of the sphere is in fact the drag force. Okay, you see, this is the drag force, and it shows 1.27 times 10 to minus 1 piconewton. Well, what about the DNA force? DNA force is this one. Well, it must show the same amount of force, but it's in the opposite direction. Why? Because the sphere is not moving. So these two forces must balance each other so that there is no motion of the sphere. Okay, this is how you produce some distance, some relocation of the sphere by just playing with the flow speed, and by just, since we are now checked the values of the forces, DNA and drag forces, you can measure the forces by just seeing these numbers over here. What happens if you drop this flow speed? Let's make it 50. Now it was 76. Let's make it 50. So you see what happens. Since I just dropped the flow speed, that means the drag force will be smaller. When the drag force is smaller, then what the DNA does is DNA will pull toward the tag, and once these two forces, drag force and the DNA force, are equal to each other, then the sphere will stop there. Okay, so this way you can give some flow, and this flow will produce some drag force, and that drag force can be measured by just looking at the numbers over there on this sphere. And well, what is the location of the sphere? Can you measure the position with respect to some reference point? Yes, you can. For example, with respect to this point, the sphere is at like 1000, maybe 1050, 1100, somewhere between 1050 and 1100. Okay, you can either measure its location by using this larger ruler, or maybe you can use this one, but this one is out of scope because you cannot use this position measurement window, because this position measurement window can measure up to distances of 300 nanometers. But starting from this dashed line. But since way out of 300 nanometers, somewhere like more than 1000 nanometer, so it is out of the scope of this, so you cannot use this one anymore to measure the location, but you can use this larger ruler anyway. Okay, so what you are going to do, let's go back to the data table in your report. What you are going to do, in fact, if you give flow speeds of smaller values like 1, 2, 3, what I shown over here, I just give a flow speed of 50 microseconds, which is large. But if you keep these small, 1, 2, 3 anyway, you can use this window more delicately to measure the position of the bed. Okay, you will see it when you play with it. For example, let's do it like maybe 5 micron per second. Now fluid flow is very small. That means the sphere will be pulled by DNA closer to this origin of your measurements. And let's see what happens over here on the smaller clear it. So it is able to measure now because the sphere is closer to the dashed line. It's less than 300 nanometers. So in the scope of this delicate measuring window, so now you can easily measure the position of the sphere by using this position measurement window, position histogram, and you can measure it by using this delicate more accurate small ruler. So even you can take a snapshot. Okay, so this is the way that you are going to measure the force on the sphere and the fluid speed and the location of the bed or the extension of the DNA anyway. So you will all record these values. I already have done some measurements, but your measurements might of course be different if you give some different fluid speeds, etc. But these are reasonable values that I just made some measurements of DNA extension and the drag force also. Okay, so you will fill out this table. It requires at least 8 data points. Okay, at least. You can do more. You can do more measurements. Let's say maybe 10 or more. And you will answer these questions. You will fill out this data table, and also you will be asked to plot. By using this data table, you will be asked to plot, DNA extension, the measurements, the position measurements, the force, and also the fluid flow. There will be 3 columns. Okay, these columns. These are going to be just plotted by using the data tool. Okay, this is the data tool. You will just put DNA location column and the fluid speed and the force. You will have 3 columns, and you will enter the data values that you measure on these columns, and you will be asked to draw some plots. Okay, so it's on the report. You will be asked to draw some plots, and there are questions, and also regarding the how to analyze this plot on these questions, etc. And you will finish the first part by working on these data collection and analyzing the plot. And on the next part, but this is part 3, this time what you are going to do is you will be doing with some Brownian motion, something with the Brownian motion. So for this part, what you are going to do is you are going to enable Brownian motion. Okay, you are going to enable Brownian motion, and by enabling Brownian motion, what you are going to do? So what you are going to do is, again, you will get some position measurements by using this histogram. Okay, let's stop it and clear it for the time being. But this in this part, what you are going to do is you are going to first give some flow speeds, but these flow speeds will be large, as large as like 250, 500, 750. You might use these 4 big flow speed ranges. Let me show first 250. Let's first give a 250 micron per second flow speed, which is very large. Okay, and first disable the Brownian motion. And just wait for the sphere set it's up in its equilibrium position, and then you will move this origin of your position measurements to the location of the sphere, to the center of the sphere. Okay, you just bring to the center of the sphere, and in order to just be sure that whether this dashed line just crossing to the from the center of the sphere, you just run your position histogram, start it, and as soon as you see the 0, that means that you're just, this dashed line is just on at the center of the sphere. Okay, as soon as you see the 0. But if if you're off the 0, you will be seeing that the position histogram will show you a non-zero value. So try to bring this to 0. Okay, now it's at 0. Okay, but you know, you can stop it and you can clear it. Now it's at 0, and what next is that you will just check enable Brownian motion. Enable Brownian motion, and then start your position histogram. Okay, once you start your position histogram, you will see that these bars, and why we're seeing these bars, because the histogram is making the position measurements continuously, and you see over here the number of measurements already. It's at like around 500 measurements now. Okay, and you know, the type of picture that you are seeing over here, if you wait for long enough time, is going to be like a bell-shaped curve. Okay, it's a bell-shaped curve, and the maximum of the curve is going to be at the 0, around the 0. The maximum of these bars that it is going to be at the 0, and you will see that since the motion of the bed is going back and forth, doing this zigzag motion, and of course, since the continuously the histogram is making the position measurements, you will see also these bars on to the left, on to the right of this center. Okay, and if you wait for some long enough time, this bell-shaped curve will be smoother and smoother. So what happens? What's going on over here? What kind of measurement are you going to do? In fact, you know, this is just to show you, in fact, it's a kind of, you know, again, position measurements, but in a more realistic scenario, there will be this Brownian motion. Okay, but you know, even there is Brownian motion, it is possible to measure the position of the bed. You might think that, you know, since the bed is just doing always this zigzag motion, but anyway, you can measure its position. When you measure its position, you know, you will produce this kind of curve, and by just looking at the center of this curve, you will be able to measure the position. Well, the bed is going back and forward on these positions, different positions, but anyway, it will locate itself the most of the time. Bed will be around the center. Okay, so well, again, as I said to you, that if you wait for long enough time, you will produce this bell-shaped curve. Well, I would suggest you to just make this around 5,000 measurements. Now it is at like around 3,600, 3,700 now. If you wait for like at least 500,000 measurements, you will have a bell-shaped curve. And I want you to measure on this picture what is the width of this distribution, because this is the kind of bell-shaped distribution. What is the width of this distribution right at the midpoint from 0 to the maximum? Okay, from 0 to maximum, find the midpoint, and right at this midpoint, just measure the width of this distribution. For example, if you just see it over here, now I'm measuring around like 0 from here, 50 nanometers, 100 nanometers, like 120-ish, like 120 nanometer. This is the width that I measure now at the flow speed of 250 microsecond. This is the width of histogram, the width of position measurements histogram in your setup. So what you are going to do next is just, you know, you just take a snapshot. You take a snapshot and just put this snapshot picture by using your screenshot tool in your computer, and copy it, and put this, or I already put below this number 12, you can put it somewhere over here. You can well, oops, let me drag it. Okay, it's picture format, yeah, square, yeah. You can put your picture to a desired position below this number 12. I already put some of my measurements, histogram measurements, but I just wanted to show it to you how to do it. So put it over there, and just under this, just type what is the flow speed. It was just 250 micro per second, right? And what is the width? I just measured the width to be 120 nanometers. Yeah, roughly. You don't need to be very accurate on this. As I said, what you are going to measure is just you measure the width of this distribution from here to here at the halfway between this on this vertical axis, at the halfway between 0 and the maximum of this bar, at the halfway, and just measure the width. So you put these, you do this like for the flow speeds of 250, 500, 750 and 1000 micron per second. Okay, you do it and put over here your screenshots. And next is just answer the questions. Okay, answer these questions, how this width is changing when you increase the flow speed, etc. These kind of questions over there, and then you will finish the homework after you just answer these questions. Well, at the end you can put the discussion. You can put the discussion. On your discussion, just type your thoughts on what you have done in this homework, on this activity, and what you have learned. Okay, so just try to discuss your results and your observations, etc. And then you will finish your homework. So guys, that's it about this second homework, which is called flow stretching and DNA force extension relation activity or homework. So that's done. I mean, that's all about that I should explain on this today's live class. Okay, so see you later.